Hello everyone. In this video, we are going to discuss about the chapter Molecular Basis of Inheritance, and this is part 13 that focuses on regulation of gene expression. So by now, we definitely know how a gene is expressed. A gene that is a component on DNA is expressed in the form of protein. And what is a protein? It is a sequence of amino acid that forms a polypeptide chain. And the intermediate in between is an messenger RNA. Okay, so this part focuses on the regulation of this gene expression, the gene that is expressed in the form of protein. And this is because, we don't need all the proteins all the time. For example, let's talk about pregnancy. A woman is pregnant only during a certain stage of her life, and all the proteins that support the pregnancy process are required only during that stage. In all other duration, these genes should be turned off. They shouldn't be expressed. Okay, so that is why we need gene expression, and every process in the body, requires gene expression. So we'll progress through this video and we'll also see what kind of questions can be asked in the NIT examination. So gene expression is a process that finally leads to polypeptide formation, and it can be regulated at various levels. Okay. So gene expression in eukaryotes is much more complicated than in prokaryotes, and by now we know this, every process, replication, transcription, translation, is always much more complicated in case of eukaryotes than in prokaryotes, because eukaryotic cells and organisms are much more complex. So the regulation could be exerted at 4 different levels. Okay, at the transcriptional level, formation of primary transcript. So here is the DNA molecule that by transcription is first converted to a pre-mRNA, that is the primary transcript. So at this level, there can be regulation of gene expression. At the level of processing, regulation of splicing. And what was splicing? Splicing was removal of introns, the intervening sequence, from the exons, that is the expressed sequence. And this process results in the formation of the final mRNA product. So, here the exons are spliced together. So, at this level also there can be regulation. Then at the level of transport of mRNA from the nucleus to the cytoplasm. And why do we need to transport the mRNA from the nucleus to cytoplasm? Because the process of transcription takes place in the nucleus and the process of formation of polypeptide chain or translation takes place in the cytoplasm. So, at this transport stage also there can be regulation of gene expression. And finally, at the translational level, there is definitely regulation of gene expression. So these are all the areas where the gene expression can take place. So there are certain important characteristics of gene expression in eukaryotes. Gene expression is dependent on the tissue in which they are found and is tissue specific. Okay, some genes are present in the heart, some genes are present in the lungs, and the gene expression is dependent on either the heart, lung, or whichever organ, and are tissue specific. So gene expression is always specific to a particular tissue. In multicellular eukaryotes, gene expression is time specific. You remember the example of pregnancy we discussed in the beginning. And specific proteins play important role at certain times. For example, in a fetus, development of an organism is coordinated by expression of several genes that are expressed only during pregnancy. Eukaryotic transcription control is generally positive because genes are usually inactive. This is again a very important point you must know. And in general, eukaryotic genes must be turned on, because the genes are inactive. Nobody likes to work. Everybody likes to be inactive, lazy, and similar are the genes in eukaryotes. Okay, so they are usually inactive and they must be turned on. Okay, so they are generally not expressed, but if you want them to be expressed, there is gene expression to turn on in these genes. Then eukaryotic chromosome structure also plays a role in regulation of transcription. How? DNA must have its protein binding sites accessible to transcription factor and RNA polymerase to initiate transcription. If you remember the part we discussed on transcription, so this was the DNA sequence, and there is something called as the transcriptional unit. And transcriptional unit has a sequence that is called as the promoter sequence. Okay, and this is the sequence, at which there is binding of RNA polymerase. If you don't remember, you can go back to that part and see the video once again. So only if this promoter sequence is accessible to the DNA can RNA polymerase bind. But if there are certain proteins that are already bound to these promoter sequence, the promoter sequence is no longer accessible. RNA polymerase can no longer bind, and transcription will not take place. So this is how basically gene regulation is there. So DNA must have its protein binding sites accessible to transcription factors and RNA polymerase to initiate transcription. If DNA is wrapped around histone proteins to form nucleosome, it has an inhibitory effect on transcription. Obviously, for example, here these are the histone proteins that are bound. RNA polymerase cannot bind and transcription cannot take place. So it has an inhibitory effect on transcription. The eukaryotic cells can also control the formation of polypeptide that are made if removal of introns is regulated during RNA processing. Very easy to understand, right? They can control the formation of polypeptide that are made if removal of introns is regulated. Okay, if the introns are not removed, the polypeptide will not be formed. Okay, or if introns are removed in a different way, there will be a different polypeptide that will be formed. For example, if two introns border an exon or exons and the introns are removed as a unit, the exon in between will also be cut out. For example, let's go back. For example, these big sequences are exons and these small sequences are introns. Okay. So what is intron? Intron will result splicing of this, and as a result the exon, in normal case the exon will be like 3 exons will be joined together. However, for example, this intron and this intron is spliced like this, there will be only these 2 exons, okay, exon number 1 and 3, and this is actually how regulation of certain genes take place. This is called as alternative splicing. Okay, so this is one of the important gene regulation processes. The polypeptide that is formed from the mRNA will be different from the one encoded by mRNA retaining those exons. Alternative splicing, as I mentioned, of transcript hence allows a single gene to code for various polypeptides. For example, this is the gene here and that is expressed in the form of mRNA. These are exon, intron, exon, intron. Okay, so these are 1, 2, 3. So in a normal situation, there will be exon 1, exon 2, exon 3, they will be together. Exon 1, 2, 3 that will code for a particular protein. However, if there is certain kind of gene regulation taking place, the splicing will take place like this, as a result of which the entire intron 2 will be excised and the final product will be 1 and 3, and this will result in formation of a protein different. For example, this is protein 1, this is protein 2. So these two proteins will be different from each other. Okay, you get it? So this is one of the other mechanisms the cells can adopt, Control of gene expression during translation depends on the time it takes until mRNA is degraded. Once mRNA is formed, it can easily be degraded. That is why we have this cap, we have a cap structure, we have poly A tail. So these kind of protect it from the process of degradation, but the mRNA eventually is degraded. Therefore, the control of gene expression during translation depends on time it takes until mRNA is degraded. Okay, if the mRNA is degraded very fast, the gene expression during translation is difficult. If an mRNA is broken down rapidly, then it cannot support many rounds of polypeptide synthesis. It can allow many copies if it remains for long. Okay, so the same mRNA can allow many copies if it has a longer life, if it remains for long, but if it is broken down rapidly, it cannot support many rounds of polypeptide synthesis. So in prokaryotes now we will talk about. In prokaryotes, regulation of gene expression can again take place at many levels. At transcriptional level, during processing of mRNA, mRNA turnover, translation level and post-translation level also. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which affects its ability to recognize start sites. Okay, so when we discussed, this is the DNA sequence and this is here P, the promoter sequence, and RNA polymerase has to bind to the promoter sequence. So sometimes what happens, in addition to this RNA polymerase, there are certain accessory proteins which affect the ability to recognize the start site or the promoter site. Okay. However, the major point of regulation of gene expression is at this level of transcription, where rate of transcription initiation is controlled. They control the rate of transcription initiation, because if RNA polymerase cannot bind to the promoter region, the process of transcription cannot be initiated. And these regulatory proteins can act both positively. If they act positively, they are called as activators, and negatively, they are called as repressors. What do you mean by positively? Positively means that these accessory proteins, let's call accessory proteins as AP, these accessory proteins can help either the RNA polymerase to recognize the transcription start site, or they can prevent the RNA polymerase from binding to the transcription start site. If they help in binding or they help positively, they are called as activators because they activate transcription. And if they act negatively, they are called as repressors because they repress transcription. So here is a diagrammatic representation of regulation of prokaryotic gene expression at different levels. Okay, this is DNA to RNA. So it is transcription. Then second is RNA processing, conversion of DNA to the final mRNA. Then is RNA stability, because this mRNA, if it is degraded very fast, then it cannot make multiple copies of protein. But if it stays for long, it can make multiple copies of protein. Then it is translation, that is formation of protein, and then it is post-translation. Now we come to the most important, where the main questions from the NIT examinations can be asked, that is operators and operon. The regulatory protein, generally repressors, interact with specific sequences called as operators. This can be a very direct question, And they will ask you, the regulatory proteins repressor, they will ask you which sequence does repressor bind to. Does it bind to the promoter? Does it bind to the operator? Does it bind to the genes? So repressor always binds to the operator. You have to learn this, Repressor will always bind to operator, which is a sequence on DNA. The operator is always adjoining to the genes whose expression it regulates. So operator and repressor are specific to each operon. Okay. So this is the structure of a normal operon. First, there is this promoter sequence. We all know what is the promoter sequence. Promoter sequence is where binding of RNA polymerase will take place and transcription will start. And then is the operator sequence, DNA where repressor binds. And then there are the genes, So these genes have an operator. So this operator will regulate expression of these genes. And what happens, now you see in this condition, the repressor is binding to the operator. So if the repressor binds to operator, what will happen? The RNA polymerase cannot bind to the promoter and transcription cannot take place. Hence, no mRNA will be formed. Okay, so this is how an operon operates. An operon consists of a promoter, it consists of operator, and it consists of genes. Okay, so operator is always adjoining to the genes whose expression it regulates. Here, this operator regulates expression of genes 1, 2, and 3. and repressor always binds to the operator. The binding of repressor to operator regulates the accessibility of RNA polymerase to the promoter region. So this is our RNA polymerase. This is the RNA polymerase. And now that operator is bound to the repressor, you see there is steric hindrance. So RNA polymerase cannot bind to the promoter. Operators are generally located between the promoter and the structural genes that they regulate. You see, this is the promoter region and this is the structural genes that regulate, and here is the operator. So operator are always located between the promoter and the structural genes that they are regulating. So this is again the diagrammatic representation of a bacterial operon. We are still talking about the prokaryotes, and the parts in green represent the operon component. Okay, so this is the promoter, this is the operator that is present between the promoter and the structural genes. So this is the bacterial DNA. Here this is the bacterial DNA and this is the regulatory gene that codes for the repressor protein. This repressor protein, once it is coded, it comes and bind to the operator, because repressor always binds to the operator. And once repressor binds to the operator, the RNA polymerase cannot bind to the promoter and expression of genes does not take place. You remember we discussed previously in this video that genes are generally in the inactive state. Now you realize what that meant. It means that the repressor is always bound to the operator, so RNA polymerase cannot bind and the structural genes are not expressed. So if we want these three genes to be expressed, we need to kick off this operator, and we'll slowly see how that happens. So since transcription is initiated at the promoters, when the repressor is bound to the operator, it sterically prevents RNA polymerase from transcribing the structural genes in the operon. I think this is very clear by now, right? So this is the RNA polymerase here that has to bind to the promoter region for the transcriptional initiation. But now that repressor is bound to the operator, there is steric hindrance, and as a result of which RNA polymerase cannot bind to the promoter and no mRNA is formed. Transcription does not take place. So a typical bacterial operon consists of structural gene, a promoter region, binding site for RNA polymerase, and operator region, the binding site of repressor, regulatory region. This is again important question from the point of view of examination. So it contains promoter where the RNA polymerase bind, it contains operator where the repressor binds, and it contains the structural genes. The regulatory gene encodes the repressor. Okay, so there is this regulatory gene that will code for this repressor that binds to operator. And then some examples of operon are lac operon, trip operon, his operon, val operon, ara operon, etc. These are examples of operon. Lac operon controls lactose, trip operon tryptophan, histone, valine, and so on so forth. And now we will discuss regulation of the lac operon. Okay, so this is a very nice model that has been developed to understand how regulation takes place in prokaryotes. So the lac operon. The operon model explained the regulation of genes required for lactose utilization in E. coli. Okay, lactose is what? Lactose is a sugar. Lactose is the substrate for enzyme beta-galactosidase and it regulates switching on and off of the operon. Hence, it is termed as an inducer. Okay, so lactose is the substrate for enzyme beta-galactosidase and it regulates switching on and off of the operon. So lactose is controlling the switching on and off of the operon and therefore it is called as the inducer. Okay, so some important characteristics of lac or lactose operon are as follows. It consists of a cluster of genes that regulate production of the enzymes needed to degrade lactose in bacterial cells. Okay, so we discussed, an operon contains what all? It contains a promoter, operator and structural genes. So it consists of cluster of genes that regulate production of enzymes needed to degrade lactose in the bacterial cells. And oxidation of lactose, which is a disaccharide composed of glucose and galactose, can provide the cell with metabolic intermediates and energy. how does lactose provide energy, So lactose can be converted by the enzyme beta-galactosidase into glucose plus galactose. Okay, and we all know glucose is the source of energy. Okay, so whenever E. coli has glucose, this lac operon will be off, then of course we don't need more glucose. But when the glucose is less, then this lac operon comes into play, and then lactose is converted by the enzyme beta-galactosidase into glucose and galactose. We'll see that very soon. So, again, like it contains cluster of genes that regulate production of enzymes needed to degrade lactose in bacterial cells, and oxidation of lactose, you can see, is converting it into glucose and galactose. So, this is lactose here, it is a disaccharide. It contains galactose and glucose, and the enzyme that causes this conversion is beta-galactosidase. Okay, so this is important, you must understand this. And then glucose is the source of energy. Then it consists of promoter, operator. We know every operon consists of a promoter and operator. And then regulatory gene, the regulatory gene is called as I gene, and the structural gene is Z, Y and A genes, which are expressed only in the presence of lactose. So what is I gene? I gene is the gene that codes for the repressor. Okay. So it will code for the repressor because it is the regulatory gene. And then there are structural genes, that is Z, Y and A, which are expressed only in the presence of lactose, So what happens, when there is increase in level of lactose, only then they are expressed. Now we'll go through each of these genes. So I gene, it is a constitutive gene. What is a constitutive gene? Constitutive gene is a gene that is always expressed, as it is always undergoing protein synthesis to produce more repressor protein and is not controlled by the promoter. Okay, so there is no promoter region for the I gene. So that is why it is constitutively expressed. It is always expressed. And you remember we discussed that genes always like to be in the inactive state. Everybody likes to rest all the time. Nobody wants to be active all the time. So these genes, the I gene is the repressor gene that maintains the lac operon always in the inactive state, which means that the structural genes are not synthesized. So I gene is a constitutive gene that is always undergoing protein synthesis to produce more repressor protein and is not controlled by the promoter. Then is the Z gene. It codes for the enzyme beta-galactosidase that hydrolyzes disaccharide lactose into monosaccharide glucose and galactose. We already know this. The first step in catabolism of lactose is the hydrolysis of the bond, which is the beta-galactosidase linkage that joins the two sugar. And this step is catalyzed by beta-galactosidase, So lactose is a disaccharide that consists of galactose and glucose. And in between galactose and glucose is a bond that is beta-galactoside linkage. So from beta-galactoside linkage, we need to break this linkage, and therefore this enzyme beta-galactosidase. So this enzyme beta-galactosidase that is expressed by the Z gene will convert lactose into glucose plus galactose. Okay. Then is the Y gene. Y gene codes for the enzyme beta-galactoside permease that increases the cell permeability to beta-galactosidases. So permease comes from the word permeability. So this gene will code for the enzyme beta-galactoside permease so that the cell is more permeable to beta-galactosidases. And if the cell is more permeable to beta-galactosidases, what will happen? Lactose will convert to glucose and galactose, and glucose will provide energy to the cell. Okay, so for this slide, the I gene will code for the repressor that goes and binds to the operator. Z gene codes for the enzyme beta-galactosidase that converts lactose into glucose and galactose, and Y gene codes for beta-galactoside permease that increases the permeability to beta-galactosides. This is important, you have to learn it. There is no other option. Then is the A gene. A gene codes for the enzyme beta-galactosidase transacetylase, the biological role of which is unknown. We don't know, what is the role of this enzyme, but we know that this A gene will code for this. So you should remember the name of the enzyme. The products of all the three structural genes are required for lactose metabolism. Generally, the products of all structural genes in an operon are required together for functioning in the same or related pathway of metabolism. You see all these three enzymes, beta-galactosidase that will convert lactose to glucose and galactose, then beta-galactosidase permease that will increase the permeability of beta-galactosidase. And then it is beta-galactoside transacetylase, the function of which is not known, but it is still related, it is beta-galactoside transacetylase. So all these genes in an operon generally function in same or related metabolic pathway. Then glucose is the preferred carbon source. However, when it is absent and lactose is available in the growth medium, the enzyme permease transport it into the bacterial cell. Okay, that is why we need permease. So what is generally happening? E. coli generally grows in colonies on a particular medium. For example, let's say this is the medium and this is the E. coli, multiple E. coli growing in the form of colonies. So in general, what is the case? In general, the medium will have glucose. And when it has glucose, glucose can directly produce energy. And in such a situation, the lac operon is off. Okay, you must know this. So when glucose is present, the lac operon is off. And why is it off? Because we don't really need to break lactose. Because anyways, we have high amount of energy from glucose. So high glucose, lac operon is off. However, when the levels of glucose decrease, then there is activation of lac operon. So the lac operon is on. Okay, why? Because here there is low glucose. If the lac operon is on, the enzyme beta-galactosidase will be active and lactose can be converted to glucose plus galactose, and then glucose will provide the energy. Okay, so this is very important. Just go through it 1 or 2 times and you will understand. It is the basic concept that you have to understand, that when there is high amount of glucose in the medium, we don't need lactose. So the only purpose that lactose is serving is that it is providing energy by breaking itself into glucose and galactose. But already when we have enough glucose, we don't need it. Therefore, the lac operon is off. However, when the levels of glucose, when all the glucose in the medium has been used up, the levels of glucose go down and then the lac operon is turned on. And then the enzyme beta-galactosidase will convert lactose to glucose and galactose, and glucose will provide the required energy. And after so much of text that was quite overwhelming, we will now see everything in the form of a diagram. Okay, so now here is the I gene. What is the I gene? The I gene codes for the repressor. So this repressor protein is coded by the I gene that is constitutively expressed. Okay, so this I gene is constitutively expressed and the repressor binds to the operator. This is very important. The repressor always binds to the operator. And if the repressor binds to the operator, the RNA polymerase cannot bind to the promoter and no transcription will take place and the lac operon is off. Okay, so this is in the absence of the inducer. So repressor binding to the operator region and preventing RNA polymerase from transcribing the operon. And this is, when there is high amount of glucose already present. So then in the presence of inducer. So what is the inducer doing? When there is inducer, this inducer goes and binds to the repressor protein and inactivates it. Very simple, So this inducer, when the concentration of inducer is there, when the inducer is present. And when is this inducer present? When there is low level of glucose. So this inducer will detect low levels of glucose. It will go and bind to this and it will inactivate the repressor. Okay, so this is very important. And once the repressor is inactivated, it does not bind to the operator, and the promoter region is free. And now the RNA polymerase can bind to the promoter region and cause transcription of the structural genes. And what are the structural genes that the mRNA codes for? It codes for beta-galactoside that will now convert lactose to glucose plus galactose, and this glucose will now provide the energy that is needed. Then it is permease, it will increase the permeability for this enzyme, and then it is transacetylase, the function of it which is not known. So this is how it acts, very simple. Like probably by now I hope that you have understood the concept, and just remember this diagram here, and here the lac operon is on. So the major thing that you need to know, when there is high amount of glucose, we do not need the lac operon because we do not need lactose. But when there is low levels of glucose, we need lactose. So the lac operon is turned on because low levels of glucose will activate the inducer, which will inactivate the repressor, and therefore repressor will not bind to operator, and therefore RNA polymerase can now bind to the promoter and cause transcription and translation of the genes that will ultimately help in utilization of glucose from lactose in the form of energy. So this is the lac operon, and that is how regulation of gene expression takes place. Okay, so this is the most important part also from the point of view of NIT examination, because questions can be asked in any form. Okay, they can ask you the enzymes the structural gene encodes for. They can obviously ask you where does the repressor bind. Repressor binds to the operator. They can ask you when there is high glucose level, the lac operon is on or off. They can ask you when the inducer is present, the lac operon is on or off, all these things. So if you understand the basic concept, these questions will be very easy to answer. Thank you for watching the video.